Thanks, Ben. Good evening, everyone. I'm Ed from Codex Research. So here we are in the third decade of the 21st century, and of course we have found cures for all the major diseases, right? What? Why not? What's happened? Well, it doesn't help that current medical research methods are really little more than tweaks of techniques we've been using for hundreds, even thousands of years. And we have learnt a lot using these techniques. But as we discover the real complexity of human biology and try to explore personalised medicine, we really need a new way to do biomedical research. Think of cells in a petri dish as a 2D experiment. The cells are aware they're in a foreign environment and often behave very differently than the cells in your body. Recently, researchers have been working on 3D techniques, usually involving bioprinters or clumps of cells called organoids. This isn't good enough. The human body is a 4D environment. Many types of cells in complex 3D structures, plus the dynamic pulsing, pressure, flow, and mechanical stress that your cells experience. Animal studies do provide this dynamic environment, but these studies are expensive, they're slow, they're ethically objectionable, and increasingly irrelevant. 92% of drug trials that succeed in mice fail in humans, reflecting just how poorly we really understand human biology. Codex Research is developing a new technology that will better mimic the human body, allow us to do away with animal experiments, and is easy to use and cost-effective. Our tech involves incorporating a smart pump system with a tissue-like scaffold within which we can grow human cells in a realistic environment under pressure and flow, just like the cells in your body. These cells can be generic for basic research or cells from a patient for truly personalised medicine. With the help of $2.5 million of government funding, we have achieved proof of concept lab results that clearly go beyond anything that can be replicated in a petri dish. In our lab prototypes, we have grown smooth muscle, fibroblast and endothelial cells in combinations that look like human arteries and shown that they begin to produce collagen. Grown clusters of cancer cells, a starting point for research into secondary tumour formation. And observed spontaneous self-assembly of cells into channels within our scaffolds, a basis for researching angiogenesis. This is currently a fundamental roadblock to the development of tissue engineering and regenerative medicine. Our technology will allow researchers to push back the boundaries of what we know about human biology and move the needle on that list of diseases that we know how to cure. We do have competitors. Their devices are expensive and difficult to use, and none can accurately replicate pressure and flow conditions found in actual human patients. Our business model includes hardware, but also, importantly, the sale of consumables. We aim to capture 5% of a $14 billion market by 2030. The advantages of 4D technology over other forms of cell culture and animal experiments will drive rapid growth in this market for many years. We are a group of passionate scientists and engineers dedicated to developing this technology. The team at the Charles Perkins Centre, our primary workspace, just up the road on the Camperdown campus. My key people, Drs. Stephanie Helder and Pearl Lee, and our close academic collaborators in Bioscience, Associate Professor Steve Wise and Drs. Anna Waterhouse and Jelena Renyak-Kovacina. I have devoted 10 years of my life and invested more than $1 million of my own savings in this venture. I have been working with and funding our academic collaborators for 7 years. We began developing the actual technology 3 years ago, amalgamating earlier work done by myself, my teammates, and the academics over the last decade. We are in this for the long haul. To advance our project, we're looking for $1 million in funding and a COO, a CTO, and a CMO to join our team. Thank you.